Hello, tonight we're going to talk about basic chemistry. A bunch of this I think will be a review from your chemistry class, and then we'll get into some new information in just a short time. Atoms are elements of a pure substance that only contain one type of molecule, one atom. They're the smallest particle that has properties of that element. Okay? In an atom, you have the nucleus with protons and neutrons, and you have electrons buzzing around in electron shells. The shells surrounding the nucleus, the first shell contains 2 electrons, and every shell after that, if it is an outer shell, can contain 8 electrons. Now, there are shells that have more than 8 electrons, and those are shells that are interior. The second or the third, fourth, fifth, sixth, and seventh shells can hold more than 8, as long as they're not an outer shell. Okay, outer shell electrons have a special name for them. They're called valence electrons because those will be the ones that are going to be involved in bonding with other atoms. There are several types of bonds we're going to talk about. The first one are called ionic bonds. Ionic bonds are formed between a metal and a nonmetal, and the electrons are transferred between the atoms. So, for example, sodium has 1 valence electron, chlorine has 7 valence electrons. Each atom wants to have an outer shell that has 8 valence electrons. It's called the octet rule. Atoms are most stable when they have 8. So sodium wants to get rid of its 1 because then the shell that's just inside that has 8. So it would have 8 valence electrons. Chlorine wants to gain 1 because if it gains 1, 7 plus 1 makes 8. So it would have 8 in its outer shell. So it wants to gain 1. So to do that, sodium gives 1 electron to chlorine, and then they're bonded together. They're held together. That's an ionic bond. Covalent bonds are formed between 2 nonmetals or nonmetal and hydrogen. What happens with the electrons this time is that they are going to be shared. Okay, sometimes they're shared equally, and sometimes the electrons are not shared equally. We'll get into that. But if they are shared, which means each one again wants to have 8. So here's 2 oxygen atoms. This oxygen atom contained 6 valence electrons, and this one contained 6 valence electrons. So sometimes this oxygen will give 2 electrons to this atom, so it can have 8 valence electrons part of the time. And part of the time, this one gives 2 to the other one, so it can have 8. By sharing them, they create a covalent bond. Now, there are 2 types, like I said, if they're shared equally or not equally. If they're shared equally, it's called a nonpolar covalent bond. If they're unequally shared, it's called a polar covalent bond. Polar covalent bonds will have, because they're not shared equally, will have one end that tends to be more negatively charged more of the time, and one end that tends to be more positively charged more of the time. So, for example, here's a water molecule. Oxygen has 6 valence electrons. Hydrogen has 1. It only has 1 shell. So hydrogen wants to have 2 electrons buzzing around. Oxygen wants to have 8. So it borrows 1 from each of the hydrogens more, and then by having 8 valence electrons, it has 2 more negatives than positive protons, so it tends to have a negative charge. Well, it has those 2 electrons more often than the hydrogens have them. Hydrogens rarely have them, but they do a little bit. Because the hydrogen doesn't have its electron, it has a positive charge because it has 1 less negative. So this end is positive, these ends, and this end is negative. It's a polar covalent bond. A nonpolar covalent bond is when they are shared equally and they don't have charged regions. Molecules that are nonpolar are things like fat, oil, gasoline. Things like that. Things that don't dissolve in water. Waxes, different things. Okay, that's your basic chemistry part. Now we're going to talk about 4 major types of molecules found in all living organisms. The first are called carbohydrates. These are different types of sugars. All carbohydrates contain carbon, hydrogen, and oxygen atoms in various amounts. Each type of carbohydrate has different amounts of those. There are lots of different types. We're not going to go into all the different types, but things like glucose, ribose and deoxyribose, those are found in RNA and DNA. Fructose, sucrose, lactose, maltose, starch, cellulose, glycogen, glucagon. Okay, there's a whole bunch. These are called simple carbohydrates. They are monomers, which means they're one single carbohydrate molecule. These are have 2 monomers connected. So, for example, maltose is made from 2 glucose molecules. Sucrose is made from 1 glucose and 1 fructose. Okay, those are disaccharides. The last group are called polysaccharides. They are made from many of these. So, for example, starch is made from hundreds of glucose molecules. To go from a monomer to a polymer, you have to connect the atoms together. So there has to be a reaction. The type of reaction that connects the 2 monomers is called a condensation reaction. So this glucose molecule and this glucose molecule are going to connect and form maltose. Okay, so what happens is in this condensation reaction, the OH on this side and the H on this side are going to connect and create a water molecule that will leaving this bonded to that oxygen right here. And so those 2 molecules will then be bonded together. That's a condensation reaction. You're giving off water, like condensation on the outside of a glass. It's given off. Okay. Hydrolysis is the opposite. Hydro means water. Lysis means to cut. So I'm going to cut a sucrose molecule into a glucose and a fructose. Remember I had that oxygen that was left in the maltose? Okay, right there. It's there in sucrose too, between these 2 molecules. So I add water to the reaction. The water is cut, lysed. So an OH goes here and H goes there. And as a result, you break them apart and you have glucose and fructose. That's hydrolysis. There are 2 types of glucose. 2 types of glucose molecules. Let's look at the difference between them. Okay, look closely. What do you see as the difference between those 2 molecules? It's a very subtle difference. Okay, hopefully you notice the OH and the H are upside down on this one and this one. Okay, this is an alpha glucose molecule, this is a beta glucose molecule. Because of that subtle difference, your body is able to digest alpha glucose. We're able to go through hydrolysis and break down alpha glucose molecules. Beta glucose molecule is found in things like cellulose, and your body is not able to break down cellulose. You're not able to go through hydrolysis with cellulose and break it apart. So we can't digest beta glucose polymers. We can digest alpha glucose polymers. You can digest starch all you want. You can't digest plant cell walls. Okay, so does it have a result? Absolutely. I mean, you can't digest cellulose. You can digest plant material. It makes a difference. And so when we're looking at various molecules, if I say, oh, look, that's a beta glucose molecule, you should start thinking about how your body reacts to that. Okay? Even termites are not able to digest beta glucose. Well, you think, wait a minute, termites eat wood. How can they digest it? They actually have a little protozoan in their intestines that digests beta glucose and can eat it for them, and then they can eat the wood. It's a little mutualistic relationship going on. Okay? We just can't digest it. So here's the structure of cellulose. Each of the shapes is a glucose molecule, and it's beta glucose. Okay? The strings of glucose molecules are held together not only by hydrolysis, a condensation reaction, in between creating the oxygens, like that. Each of those was condensation. But it's also got hydrogen bonds between, holding them together on the sides. So it forms these very straight lines of glucose molecules. Okay, the next major type of molecule is called a lipid. Lipids are things like fats and steroids. They are not water soluble. They are nonpolar. Okay? One interesting thing, all 4 major molecules, except lipids, have monomers and polymers. Monomers being the basic unit, polymers meaning multiples of the monomers. Lipids don't. They're all very different molecules, but because they're all nonpolar, they're all put in this major group. Some have a glycerol and fatty acid chains, but not all of them. So they just really don't have monomer polymers. Here are some examples, and as you can see, the shapes of them are very, very different. This is a triglyceride. Have you ever heard of triglycerides in your bloodstream? It's one of the fat molecules that flows around your body. You need some, you just can't have too much. Steroids, estrogen, progesterone, testosterone, also things like cholesterol are steroids. And phospholipids. Have you ever heard of a phospholipid? Phospholipids are the major component of all cell membranes. They have a hydrophilic head region, and then they have 2 legs that come off. Now, one of them is always bent. Now, you say, why is that bent? What happens here, each kink is a carbon molecule. See right here? Carbon, 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 carbon. It's just like that. And coming off from each of the carbons are 2 hydrogens, except for right here. Here we have 2 carbon bonds. That means 2 electrons are being shared between the 2 carbons. As a result, it's missing 2 hydrogens here, one on either side of that. So it makes it kinked. It just bends it. Changes the shape of the molecule. But all phospholipids have that kink. When you're looking at molecules, what you can do, you look at this and you say, that looks kind of weird. Look up here where it's highlighted. That's a phosphate group. So we know in phospholipids they have a phosphate group. Lipids is going to refer to the 2 tail regions where you have carbon, hydrogen all the way down. Right here. Okay? Anyway, those are examples of lipids. The next major molecule are called proteins. They do have a monomer. The monomer of a protein is an amino acid. There are 20 different amino acids in your body. I think of them kind of like letters in the alphabet, and I'll tell you why here in a minute. Amino acid has all the same basic shape. They all have this blue part region identical. And then where this H is coming off, this is called the variable region. It's called an R group. Each of the R groups has a different shape. They're different atoms that are attached there. And that's what makes them all different. These 20 amino acids are going to build every single protein in your body. And just like you have 26 letters in the alphabet that can build thousands and thousands of words, these 20 amino acids can build thousands and thousands of different proteins. The order of the amino acids will build the exact protein. Okay, so that's kind of how you relate it in your mind. There's lots of different types of proteins that can be found in your body. Anything from structural proteins like spider silk, storage proteins like egg white or albumin, transport proteins like hemoglobin carrying oxygen, hormonal proteins like insulin or estrogen, receptor proteins that receive neurotransmitters in your nerve cells, contractile proteins like actin and myosin in your muscle cells. Antibodies are proteins in your immune system. Enzymes, every enzyme that runs every single chemical reaction in your body is an enzymatic protein. So there's several of them. Now, to build a protein, you have to create a bond, and the bond is called a peptide bond. To create the peptide bond, you go through, oh, look at this. Does it look familiar? OH, H, giving off water. Yep, that's a condensation reaction. A condensation reaction resulting in a peptide bond. That's important. So to put them together, you have to go through condensation, you give off water, those two then attach, and you've attached another one. If I want to break a protein apart, what type of reaction are you going to go through? Hydrolysis. Remember? Hydrolysis, hydrolysis, so you're going to add water, you're going to lyse it, break it, and then you can break that off. Hydrolysis. Okay? Now, as you can remember, they all have the same basic shape. Here's the same shape on each of them, but you see the different side chains, the R groups, how each one is different. Okay? They are all different. Oops, wrong direction. Okay, so if you look at the structure of a protein, there are 4 levels of structure that we're going to look at really quick now. The first is the order of the amino acids that build the protein. So what order are these going to be in right here? What order are the letters of the alphabet? Okay, so the amino acids, like I said, there are 20 of them. Proteins can be anywhere from 50 amino acids long to 1,500 amino acids long. They vary in length depending on what they're going to be doing. But the order of these letters, these nucleic acids, amino acids, the order of the amino acids will determine what protein you have. As you can see, there are, if you actually count it, there are 20 of them. Okay? Each one is held together by a peptide bond caused from a condensation reaction. Okay, so that's your primary structure. Okay, peptide bonds hold them together. The secondary structure takes those amino acids, that chain, and it does one of two things. It either will take that chain and coil it around, forming an alpha helix, or it folds it like a fan, and that's called the beta-pleated sheet. Those two different types are called secondary structures, and the bond that holds those together is a hydrogen bond. There's the word right there. Those dots represent a hydrogen attaching to an oxygen, forming a hydrogen bond. Okay? The tertiary structure is next. The tertiary structure holds the secondary structures together. So the purple line here would be that secondary structure here. It'd be the coil, the alpha helix, or it'd be that beta-pleated sheet. Oops, wrong way. So here's that coil or beta-pleated sheet. Those side chains that come off now come into play. Well, one of the molecules, I think it's cytosine, has sulfur coming off from it. Not cytosine. Which one is it? I don't remember which one it is. I'd have to look it up. But it has sulfur coming out of it. Because there's a sulfur, sulfur is attracted to other sulfur molecules. And so anywhere along here that has another sulfur is going to be attracted to that one, and it's going to cause this to fold back over on itself, and those sulfurs connect, forming a disulfide bridge. There's lots of different types of bonds in the tertiary structure. So disulfide is one. You can also have hydrogen bonds. You can have hydrophobic interactions where these guys hate water, so they're going to be attracted to each other so they can repel water. You can have van der Waal interactions. You can have an ionic bond or a covalent bond forming. So there's lots of different reactions in this tertiary structure that are helping to form the three-dimensional shape of your protein. Now, many proteins stop at the tertiary structure, and it now has a three-dimensional shape that will perfectly fit another atom, whatever it's going to react with or whatever shape it has to have. This tertiary structure gives it to it. Some proteins, though, are built from more than one protein molecule. And if it does, it's called a quaternary structure. This particular quaternary structure is hemoglobin. It's built from 2 alpha helix chains down here in the pink and purple and 2 beta hemoglobin chains. Each of those shapes is a tertiary structure. 4 of them come together, 2 alpha and 2 beta chains, come together to create hemoglobin. Okay, each one of these, by the way, is the heme group. That's where the iron is. It's going to carry the oxygen. Hemoglobin can carry 4 oxygens. So just to review, primary structure is the order of the amino acids. That can either be pleated or it can be a helix as a secondary structure. Those then get folded and bent into a three-dimensional shape. And then if several of the three-dimensional shapes come together, you can form the quaternary structure. The last type of organic molecule is called a nucleic acid. These are examples include DNA, RNA, ATP. They're all very, very important molecules that you have to have in your body. The monomer of each of these is called a nucleotide. A nucleotide is made from a sugar, either deoxyribose or ribose sugar. Each of those, deoxyribose and ribose, are called a 5-carbon sugar because they have 5 carbons in them. It's shaped kind of like a pentagram or a house. They have one of the sugars bonded to a phosphate group, and then one of 5 different nitrogenous bases. DNA and RNA are the 2 most common. So let's look at their structure first, and then we'll talk about ATP. DNA has the sugar deoxyribose. RNA has the sugar ribose. DNA is a double helix, and it's 2-sided. It's like a twisted ladder, and you've got 2 sides to your ladder. RNA is like you took a chainsaw to the ladder and cut straight down. You have half a ladder. Okay? Single-sided. The nucleic acids found in DNA are adenine, cytosine, guanine, and thymine. The ones found in RNA are adenine, cytosine, guanine, and a new one, uracil. So you have to remember which one has thymine and which one has uracil. Examples of DNA. Examples of RNA, there are 3. rRNA, which stands for ribosomal RNA, and it makes up part of a ribosome. Transfer RNA, which is clover-shaped, and messenger RNA. And we'll find out a lot about RNA when we talk about protein synthesis. So let's look at that nucleotide structure. You have that pentose sugar, you've got one of the nitrogenous bases, and you've got a phosphate group. And you can look at the difference between DNA, deoxyribose, and ribose found in RNA. And what you're going to see is that right there. That's your major difference. Okay? Look at the difference in the nitrogenous bases. Cytosine, thymine, and uracil have a single ring, and there are slight differences between each of those. I'm not going to make you memorize them. Purines, though, have a double ring. Okay? So here's your DNA molecule. To help DNA make sure mistakes aren't made, when we're going to copy the DNA, adenine and thymine always go together. And if you look at them, adenine and thymine, these guys are always going to go together. When they go together, the hydrogens are going to form hydrogen bonds. A and T form 2 hydrogen bonds. Cytosine and guanine are going to form 3 hydrogen bonds. And because a 3 doesn't match up perfectly with a 2, adenine will never bond to cytosine or guanine, and thymine will never bond to cytosine or guanine. They're only going to bond to the one they're supposed to bond to. At least that's the way it's supposed to happen. We'll leave it at that for now. Okay, 2s and 3s, and hydrogen bonds hold them together. The last thing we're going to talk about, I think, is ATP, the structure of an ATP molecule. It has ribose sugar, it has adenine, and it has 3 phosphate groups. That looks almost identical to the regular nucleotide. Okay, it had the ribose, the phosphate group, and adenine, but this time it has 3 phosphate groups. ATP is extremely important because what it does is it is the energy storage molecule in your cell. When the cell needs energy, it breaks off one of these phosphate groups, and that releases a whole bunch of stored energy in this bond right in here. That energy is released, and the cell can use that energy to do whatever it needs to do. If the cell makes energy, like during cellular respiration or photosynthesis, if it's going to make some energy, what it does is it takes a ADP, adenosine diphosphate, di means 2. It has 2 phosphate groups. It takes one of those and attaches a new phosphate group to it, and by doing so, it takes a bunch of energy to do that. That energy is stored and can be released when the cell needs it. So this is a really good example of how the cell uses and reuses molecules. Okay, this is an ATP molecule, and that is it for today. No homework with this PowerPoint. Have a great night.